Hi everyone and welcome to this video. In this video, I will show a complete drug docking simulation in the context of Alzheimer's disease. In silico studies are very important for screening drug candidates. It can save years in the drug design pipeline by narrowing down the list of viable drugs and thus bringing down the overall cost of drug development. As most of us know, Alzheimer's disease is a neurodegenerative disorder that affects cognitive function. One of the key mechanisms of the disease is the formation of amyloid beta plaques. As shown in this figure, fragments of the amyloid precursor protein are abnormally cleaved, leading to accumulation of the amyloid beta peptides. These peptides clump together to form plaques in the brain, which disrupt communication between neurons and trigger immune responses and inflammation. The PET scans show healthy brains at the top and diseased brains at the bottom, with the red indicating plaque formation. As shown in the red text, different treatment approaches target different parts of this life cycle. In this study, I will be focusing on lecanemab, which is a monoclonal antibody that targets amyloid beta protofibrils. Modeled after IgG antibodies, the Fab region of lecanemab binds to the amyloid beta protofibrils, while the Fc region engages microglia, that are immune cells of the nervous system, for clearance of the plaque through phagocytosis. This is an FDA-approved drug which slows down cognitive decline. We will model the interaction of lecanemab with amyloid beta plaques using a molecular docking tool like HADDOCK. Before we can use docking, we need the folded structures of both amyloid beta and lecanemab. Luckily for us, the amyloid beta structure is readily available in the protein data bank as 8AZS. This is shown here. Unfortunately, the structure of lecanemab is not available in the PDB. This means we have to use protein folding tools to get the structure. We will do this through AlphaFold. This is the Google Colab that implements AlphaFold2. However, to use this, we first need the amino acid sequence for lecanemab. This is available through DrugBank as seen here. As we can see, there's a heavy chain and a light chain. AlphaFold expects multiple chains to be input with a colon separator, so we do that. Since this takes a long time, I've kept a completed run here. As we can see, the structure is iteratively refined. The PLDDT coloring has blue for high confidence and red for low confidence in the prediction. And we get the final structure here, with plots of performance. As expected, this shows the nice Y-shaped structure that is typical of immunoglobulin antibodies. I have downloaded the results file here, which contains the PDB of the final fold. The file with rank 001 is the best fit, so we will be using this in HADDOCK. There's some post-processing needed on the PDB file, which I am not covering in this video, but after running those scripts, we have a file here called lecanemab-clean.pdb. We will use this. So now we have the two PDBs for amyloid beta and lecanemab. Now let's go to HADDOCK. Let's submit a new job. Here, I will use lecanemab as the large protein and amyloid beta as the small molecule ligand. Other than the PDB structures, the key parameters HADDOCK needs are the chain IDs and binding residue locations. I have already run the simulation with these parameters and saved them up in a JSON file here. Again, this simulation takes quite long, so let me switch to a completed run. The simulation results are shown below. The top cluster is the best scoring cluster, which has the minimum or most negative score. The plots below show the energy plots and various other metrics for the simulation. I'm not going into their details in this video. If we visualize the docking, we see that it shows a good and tight fit in the Fab region of the antibody. We can now use this drug-protein complex PDB file to analyze further properties, such as its stability in the cerebrospinal fluid where this has to act. That is the subject I cover in another video. Hope you found this useful and thank you for your time.